Hello, this is Jacob Bartholomew, the creatively deficient mind behind the Biochemistry Creative Response Sock Puppet Show. Without a doubt, the concept of biochemical regulation is the coolest thing that makes life possible because we can see shadows of the same problems and solutions at the organismal level. To begin our demonstration, we should begin with a fundamental question. Why is biochemical regulation necessary? Why can't every button be pressed at once at the same time? The answer to that question is teleological, and to demonstrate, all I need is a zero-turn lawnmower. If my goal is to cut the grass in front of me, the most effective, least wasteful method is to direct both handles to the forward position. This process would be catabolic and irreversible. The lawnmower can be considered our enzyme catalyst. Without our lawnmower, which positions the blades in the best location, best speed, and best width, cutting grass with my teeth would be quite difficult and time-consuming, though it would be possible at a much slower rate. It is worth noting that the same amount of energy required to cut the grass with a lawnmower as it would be to bite the grass off or to cut it with a small pair of safety scissors. But the lawnmower uses combustion from gasoline to turn its engine, wheels, and blades instead of calories broken down to form ATP, powering myosin ATPase in my muscles. If the same amount of work is being done, the same amount of energy is required to do that work. But back to our riding lawnmower, what if instead of pushing both handles forward, I pulled one back, simultaneously directing the lawnmower forward and backwards? This zero turn will rotate in place, consuming gasoline for no good reason, cutting grass that has already been cut. A more precise analogy would be if I was cutting each blade of grass with a pair of scissors and a friend was coming right behind me, restoring each blade of grass in an anabolic process. How many more calories are wasted effectively doing zero work in this situation? This would be considered a futile cycle. In glucose metabolism at the cellular level, this process is tightly regulated, especially by phosphofructokinase. Glucose metabolism, intermediating glucose and pyruvate levels, is regulated in quite an analogous way to eating is at the organismal level. When properly regulated, in the simplest terms, there is no reason to spend your entire paycheck on as many apples as your paycheck can buy. There is no reason to eat all your food at once. You're constantly feedback responsive to prevent waste. Things start to change with more separation and specialization, though, as seen in eukaryotes, particularly those which are highly developed with organs. If I bake 24 cakes in an anabolic process, I might eat 2 of them for myself in a catabolic process and ship the rest to people that might need those cookies more than I do, in exchange for things necessary to make more cookies. This, at the level of an individual, is futile, wasteful, and reckless. But because this cookie baker is exchanging with others, a kind of division of labor becomes mediated. In humans, this kind of interaction happens between the liver and skeletal muscles. The liver plays the role of the baker, and they are constantly producing glucose in excess of its own cellular demands. And the skeletal muscle plays the role of the cookie customer, exchanging lactose for glucose, and the liver using that lactate for gluconeogenesis. This is called the Cori cycle, and it is a great example of the massive benefits of a futile cycle. So then a better question reveals itself. At the cellular level, a futile cycle is usually avoided, and at the organismal level, a futile cycle can sometimes be of great advantage. What is our invisible hand of the market? This is where biology is so much better than human organization. Our invisible hand is not some mechanism prioritizing certain cells over another, purely in terms of energy demands, nor is it reducible to an even distribution of cellular energy. The first option would be self-destructive. Our catabolic specialized cells cannot be prioritized over our anabolic specialized cells, or else the anabolic cells become self-sacrificing and the catabolic cells no longer have the means to exist. Our capitalists cannot exist without our proletariat. The second option would also be a failure. Particularly productive cells cannot equally consume their own labor, and unproductive cells themselves necessary to the survival of the organism, quickly starve. Thus, a multicellular organism operates according to the beautiful maxim, from each according to their ability, to each according to their needs. Each cell operates according to their own energetic self-preservative and dictated by materialism alone. And still, the miracle of the dialectics of life allow for such complexity as can be seen at this simply understood level. So again, what is our invisible hand? How is the body effectively so centralist yet responsive? The answer ultimately comes down to DNA. The structure of DNA is held together thanks to complementary base pair hydrogen bonding and beneficial base stacking as a van der Waals force. This means that by changing the nitrogenous base sequence, the forces which hold it together is also modified. This change in structure can make DNA more or less accessible to transcription, particularly AT stacking as low energy, easily accessible coding regions. The sequence of DNA, arguably much more important than playing the role of self-stabilization, is what codes for the RNA. It is worth noting that the DNA is not meaningfully independent from what it codes for. DNA requires replication in the cell cycle, and that replication is carried out by proteins, such as topoisomerase, which relieves strand tension by cutting phosphodiester bonds, helicase, which unwinds the double helix, RNA primase, initiating the coding region, single-stranded binding proteins, which prevent the DNA from reannealing, and of course, DNA polymerase to lay down nitrogenous bases. For the liking strand, an additional protein is required, DNA ligase, to seal nicked DNA. All this time, proofing proteins are involved to ensure the fidelity of replication and preventing mutations. Therefore, even DNA itself is answerable to the environment of the cell. If bases are out of equilibrium, DNA becomes unfaithfully replicated, in turn affecting the fidelity of proteins eventually synthesized. Again, it is important to remember that DNA codes for these proteins. This transcription is controlled by DNA structure, including repressors and activators, themselves having chemical affinity to sequence-specific regions. If a messenger RNA is synthesized, requiring promoter sequences in RNA polymerase, it must be modified in eukaryotes, often by small nuclear, ribonuclear proteins, into several possible protein products, because different introns can be excised for different products. This final, modified messenger RNA is a sequence of amino acids, which is finally translated into a protein through codons in tRNA mediated by ribosomes, costing roughly 3 GTP equivalents per amino acid. These proteins can be post-translationally modified, but those amino acids still aren't done. The sequence and structure of that polypeptide simultaneously directs its function and half-life. The protein is eventually marked for ubiquination, so that those same amino acid Lego bricks can be reused for alternative purpose, so that the cell doesn't constantly express every possible protein, which would be seriously energetically expensive, especially if de novo amino acid and nitrogenous base formation is required. This is the ultimate mechanism and explanation for why the cells in our eyeballs and the cells in our toes are so different, even though it is the same DNA. But none of this would be possible if cells didn't act according to the principles of communism. I could go even further and suggest unknowing altruism as the evolutionary dictated explanation for cellular apoptosis. Ultimately, cellular self-sacrifice for the sake of organismal fidelity, but that's not necessary for this discussion. It is wonderful how all of this is genuine communism lived out every day in every eukaryotic living thing at the cellular level. It's not just human nature. It's the nature of prokaryotes. It's the nature of eukaryotes, not prokaryotes, my bad, as a product of hundreds of millions of years of environmental pressure and struggle for existence. This is the beauty of biochemistry. This is what I've learned the past 2 semesters.